So thank you all for coming to this seminar. This is the second one from a series of seminars given by a young researcher from the Bordeaux Population Health Research Center. So people recently who recently joined us either as an associate professor or as a researcher, Inserm researcher. So Denis was recently recruited this year as a researcher, full-time Inserm researcher, and he will present his work about joint models. And next week we will have another one by Morgan Linard, who should be in French and who was also recently recruited in our research center. Thank you.
All right, thank you, Hélène. So I'm going to talk about joint models and cancer clinical trials. This is pretty much my PhD topic, which I defended in December 2020. And since then, I've done 4 years of postdoc to reinforce my methodological skills. And today, I want to try to make a statement about how useful joint models can be for the, cancer clinical trials data analysis. So let's start with a simple question. How do we currently evaluate if a cancer treatment works? We usually rely on randomized clinical trials, right? We need to measure something. So the endpoints, we have overall survival, the time to death from any cause. This is the gold standard. We also often have a co-primary endpoint, the time to progression of the disease. So we have those two metrics that we measure, and we compare the two treatment lines with some statistical tests, the log-rank test or Cox proportional hazards, which gives hazard ratio. So that's what we target, hazard ratio. One important point, the way randomized clinical trials work is we have a population, we have two treatments, standard of care or placebo versus the new treatment, and we just randomize people. So the population is heterogeneous. Maybe there are old people, they are more at risk of dying. But because of randomization, we expect to have as many old people in both groups, and the effect of age should be averaged out. So this is fine. It works very well. We should have an unbiased treatment effect. So those methods, they assume homogeneity. They consider the heterogeneity, the age difference, for example, just as a noise to average out. So if you look at pretty much any report of the results of a cancer clinical trial, I took one randomly on the New England Journal of Medicine, you can see two things, a hazard ratio for disease progression and a hazard ratio for death. That's the final result of the clinical trial, those two hazard ratio, that's the main result. There are sensitivity analysis, of course, but that's what makes the decision, do we accept this treatment or not? So this is standard, but sometimes we do include some baseline covariates, for example, age. Why? I just said it's averaged out, so we shouldn't need to adjust on anything, right? There are some reasons. If you look at this guidance document from the FDA, they explain adjustment remove the noise of patient heterogeneity from the error term, which increase statistical power. So when we explain the noise, when we explain, oh, this guy, he dies early, but that's because he's old, not because of treatment or anything, we explain the residual variability, and we increase the statistical power. The second point, randomization is not always perfect. So we might want to correct for imbalance. If there are more old people in one treatment group, adjusting for age will correct for this. So even in randomized clinical trials, we can get a benefit of adjustment. The last point, the most important one for today, and you have details in this Biometrica paper on the top right. In Cox models, omitting prognostic factors mathematically bias the hazard ratio towards 1, which dilutes the effect of treatment. It's called attenuation bias, and it's quite important. The more noise you have, the more your treatment will be shifted towards the no effect. That's a big deal. To try to clarify, this is directly linked to survival of the fittest, survivorship bias. If we consider heterogeneity is very simple. Either you have big tumors or small tumors. Big tumors, you have a high risk of dying. Small tumors, a low risk. In a randomized trial, the population is homogeneous at time 0. There are as many big tumors in both groups. But over time, if my treatment is efficient, the control group, you can see, the standard of care, it cannot save the big tumors. They die early because they are at higher risk. But the effective treatment might save some of them. So later, you might have imbalance in the population. And this, because we average the hazard ratio over the entire follow-up, it will shift slightly the treatment effect towards no effect. What's the solution? Simply adjust. So if we account for red or blue in our model, then we do compare treatment conditional on this. We compare the red with the red and the blue with the blue. And then we eliminate this attenuation bias. This is oversimplified. In the real world, there are many factors, and the heterogeneity is much more complex. Each patient has its own tumor size and trajectory over time, so it's more complex, but that's the main idea. Adjusting and explaining the variability increase the statistical power. So now I mentioned two outcomes. Overall survival is very simple to understand. But progression, how do we know that the cancer is progressing? When is it triggered, the progression? There are a set of rules that define what is a progression. Those are called RECIST criteria. And it's based on the measurement of the tumors. So every patient in every randomized clinical trial of solid tumor cancer will go through a CT scan or MRI scan, whatever, and the clinician will select up to 5 tumors. Sometimes there are more, but the limit is 5. And he will measure their longest diameter. So he takes the 5 biggest ones, and they are measured, and we take the sum. This gives us an information about the tumor size for each patient. And over the follow-up, every couple months, the patient goes back to the clinician, and we measure again the same tumor. So we know, does it increase in size, does it decrease? We have longitudinal information about how the tumors evolve in size for each patient. So for example, here we have a patient. He starts with a 10 centimeter tumor size, or SLD, sum of longest diameter. I will call it tumor size for simplicity. 10 centimeter, it could be 5 tumors of 2 centimeters, for example. It might decrease because treatment effect. So the tumor size shrink over time, minus 30%, and then it might reach 5 centimeters. If it increases by 20% at least, that's a progression. We trigger the progression when we reach this threshold. So you can guess we lose a lot of information. We have a longitudinal follow-up for each individual, and all we get out of it is, did it increase by 20% or not? There are other triggers for progression because those are only up to 5. So if there are more than 5 lesions, more than 5 tumors, we don't measure them, but the clinician will still compare from one visit to the next one. Did it increase or not? And if the clinician decides he sees an increase, this is a progression. Also, if new tumors appear, metastasis, that's a progression. As soon as we see a new tumor, it's not measured because we only measure the 5 that were selected at baseline, but this will trigger a progression. So we have 3 sources: measured tumors, non-measured tumors, and metastasis can trigger a progression of cancer. So as I said, there is a lot of loss of information. A patient with 19% growth is considered stable, and 21% progression. But they are very similar in practice. So this outcome, this endpoint, progression-free survival, it's very often used in practice. As a primary endpoint, co-primary endpoint with overall survival, it's used very systematically in some specific cancer. But if you look at this meta-analysis, you will see that correlation between PFS, progression-free survival, and the gold standard of overall survival is quite weak, up to 0.6. As a statistician, I can tell you, this is not good for a surrogate. We want 0.9, 0.95 for a good surrogate. So why is it used? FDA gives us an answer in some guidance for industry paper. They say PFS, progression-free survival, can represent a direct clinical benefit, not survival, clinical benefit, based on the clinical consequences of delaying or preventing progression. So PFS is accepted because delaying tumor growth and the associated symptoms is a valid medical goal. And even if it does not guarantee a longer life, you still die at the same time, but you have some sort of better quality of life. You have smaller tumors, and tumors are associated to symptoms. So they say it's fine, it's still valid, and it's still useful to approve treatment based on this endpoint. One more point, overall survival, despite being the gold standard, is not perfect. In cancer clinical trials, sometimes when we observe progression, we give a new treatment. We switch, we add treatment. this is not good because the treatment we want to test is the original one, not the one we switch after progression. So this might obscure the drug's survival benefit, and it makes overall survival not a perfect endpoint. Nothing is perfect, actually. So what about those individual tumor size trajectories? They are quite informative. For each individual, we have a trajectory of the size of tumors. So some individuals, for example, they start with a certain tumor size, and they might respond very well to treatment. The tumor size decrease. It can even reach a zero, completely disappear. Good news, that's what we want. Some patients might not respond at all to treatment, so their cancer might just progress and the tumor size might just increase, regardless of treatment. Sometimes we do observe a decrease in tumor size, but later on we have an increase, a change of trend. That's very common with chemotherapies, the development of a resistance to the treatment. The treatment works, but just for a limited amount of time. And sometimes the tumors are staying stable and the treatment doesn't change much. So we have a lot of heterogeneity that is captured by this marker. It's very informative. But where does it come from? Multiple drivers, of course, multifactorial. Some drivers are observed. For example, I mentioned the age. Maybe old people have bigger tumors, so that could explain some of the variability in tumor size. Some drivers are not observed, a specific genetic variant. One variant could be associated to a good response to treatment, and another variant could be associated to the absence of response. But we might not measure it. It's unobserved, so we don't know. Maybe that's the true reason why we do observe different trajectories. And the last, and the most important one, treatment. We expect treatment to affect the evolution of tumor size, right? So it's deeply multifactorial, but it's also a rich source of information, tumor size. Actually, why don't we just look at the tumors? Since they are a direct manifestation of the cancer, a good treatment should just reduce tumor more than the standard. If it does, then we should just approve it, right? Simple. Not really. To answer this question, I will make a small detour through some mainstream media and social media. There's a rumor about some revolutionary cancer vaccine. 3 years of trials proved safety and high efficacy. 100% efficacy in clinical trials. Wow. Millions of views, hundreds of thousands in France. This is spreading quite well. So I looked, where does this come from? There is an official statement from the Russian state agency that says we saw reduction in tumor size ranging from 60 to 80%. Wow, good, interesting. Must be a good treatment. Not really. When you look more carefully, those are only animal models, mice. There is no phase 1 trial as of today. And if you look at this paper published in Biostatistics, you will read that even after successful animal testing, 96.6% of oncology drugs fail in human trials. So we are really in early stage of research, and this is far from being what people seem to say online. The reason why I talk about it is because you cannot just look at a tumor reduction in size to conclude about treatment effect. And there is a justification for it. Especially for those new treatment, targeted therapies. Immunotherapies, you might know this one, it was Nobel Prize a couple of years ago. The point is, cancer is about some cells escaping the immune system, and they can replicate, and that's the basis for tumors. Those targeted treatments, they aim at restoring the ability of the immune system to fight those tumors. The idea is quite simple. We identify a marker on the surface of cancer cells, a peptide, and we tell the immune system, if you see this marker, just attack. And it works well. The treatment reduces tumor size. That's exactly what we want. The issue is this exact same marker is often also on healthy cells, lung cells, heart cells. So the treatment is very good at killing the tumors, but it also kills the host. So just looking at the tumors is not enough because you have good information about efficacy, but no information about toxicity. So if we only look at tumor size, the drug looks efficient in this case. But if we only look at survival, it looks like a failure. So what about a method that sees both simultaneously? I guess you see where I'm coming. Joint models. That's the whole point of joint models, modeling simultaneously a longitudinal process, tumor size over time, and a survival process, the risk of progression, the risk of death. So joint models, they treat tumor size as an evolving predictor. As the tumor grows, the hazard of death or progression updates in real time. There is a synchronization between the evolution of tumor size and the associated risk of event. And there are many advantages. Among those, informative censoring. We might never observe big tumors, because big tumors are associated to such a high risk of death that people die before we can measure them. So our tumor distribution is censored. Joint models can correct for informative censoring by the event of interest. Measurement error also, those measurements are subject to some residual error. We want to filter it out, and that's what joint models can do. Okay, so to summarize, the standard approach, what people do now for clinical trials, is about summarizing and then analyzing. We collapse the complex tumor size trajectory into a single plus 20% event. We lose a lot of information. We treat individual heterogeneity as noise to be averaged out, which results in some loss of information and a lower power. And this has consequences. More patients exposed to placebo-controlled arms, effective drugs take longer to reach the market, so this has quite an impact. On the other hand, joint models, they model the continuous SLD values over time, and they reduce unexplained variance, which increase the signal-to-noise ratio, as I explained earlier. So the idea of the joint model is to explain heterogeneity to gain statistical power. And it transforms those routine tumor measurements into statistical gain. The measurements are there, systematically recorded, any clinical trial. It's not about doing some new measurements. We take the data available and we use it in a more efficient manner. Okay, let's talk a little bit about the models of interest. So we need to model the longitudinal tumor size for each individual. SLD is my tumor size. I will use a mixed effects regression model to decompose the information from tumor size. The idea is we have fixed effects for population average trajectory. Treatment, for example. Those fixed effects will describe how tumor size evolves over time, conditional on treatment. Do we observe a difference on average between the two treatment lines? We can also have other covariates, age, gender, whatever. Random effects. They capture the individual deviation from the population average. So let's say my average is 10 centimeters, but this individual has a 15 centimeter tumor size. 10 centimeter is the value given by the average, and plus 5 will be my random effect. He's higher than the average of his group. He has bigger tumors than the average of his group. So the standard Cox model will just treat heterogeneity as statistical noise to be averaged out, and we'll only adjust for baseline covariates. So we have a treatment effect, our effect of interest. This will give us our hazard ratio, and some baseline covariates. Okay? So this is the standard approach, but now we propose the first joint model, shared random effects. The idea is to adjust for individual deviation from the population average. You remember, individual deviation, plus 5, for example, was my deviation. He has bigger tumor than his average. We compare big tumors with big tumors, small tumors with small tumors. That's the idea. We adjust for the noise, and there is no treatment effect. Treatment effect is here, in the fixed effects. So we just adjust for individual variability, and we get an adjusted hazard ratio. Joint model with current value, the idea is to link the risk to the longitudinal tumor size directly. So we take the fixed effects, the average, and the deviation. The risk of event, let's say the risk of death for individual I at time t, depends on his tumor size at the same time. So here, it depends on 5 plus 5, and here it will depend on 15 directly, the tumor size itself. Okay? Now, the quantities of interest. The final result of the clinical trial is the hazard ratio. That's what we do with the standard Cox model. Joint model with shared random effect, we do the same. A simple hazard ratio, except that this one is adjusted. So we compare big tumors with big tumors, small tumors with small tumors, after accounting for treatment effects. The last one, because we have different trajectories conditional on treatment, there is a delta, a difference between the two treatment lines in terms of tumor size. And this is taken into account in my survival model. So I end up with something time-dependent, the difference over time between the tumor size between the two groups. And I end up with a time-dependent hazard ratio, because I have a time-dependent component. So the idea is to have a treatment effect that is time-dependent on my longitudinal marker, and a time-dependent effect of my longitudinal marker on my survival outcome, some sort of mediation of treatment effect through tumors. And we also have a direct residual effect for any difference in risk between two treatment lines that is not explained by my tumors. Okay? One important point, there is this paper in the Journal of Clinical Oncology by Joseph Ibrahim. This is basically the biggest paper for joint models in cancer. It has hundreds of citations. And if you read it, you will see the longitudinal model he proposed is including treatment as an indicator with a constant effect. I think it's a pretty bad idea in a randomized clinical trial to assume a constant treatment effect. Why? Because of randomization. At time 0, the two treatment lines have the same average tumor size. They are supposed to. Treatment effect starts after time 0. We start the treatment. So at time 0, they should have the same tumor size. The two lines should be starting at the same point, and over time, we will see the treatment effect. Is the treatment reducing tumors stronger than the standard of care or not? So what Ibrahim is doing is mathematically biased towards the null. It shifts the treatment effect towards the null because of these properties of randomized clinical trials. So just to mention that, of course, we will have a time by treatment and not a time plus treatment, such that we capture the effect of treatment at baseline, which should be 0. We control for randomization, and then we have a treatment effect over time and two curves that can separate. Another important point, so we can do joint models for overall survival, but for progression, I mentioned that progression is triggered by a 20% increase in tumor size. So we cannot have tumor size in the predictor and in the endpoint. That's circularity, and that's mathematically wrong. So this is arbitrary. A bit like Ronald Fisher, who stated that any statistical test is significant at the 5% level. This is arbitrary. It works, it's fine. This threshold might capture some progression, but there is no clinical motivation. On the other hand, when metastases appear or when there is a progression that is confirmed by the clinician, this is unequivocal, no questions. So we propose an alternative endpoint to be able to do our joint model for progression, where we define an unequivocal disease progression. So we basically discard this part, and we will use tumor size in a different way as a longitudinal predictor, and we only consider progression when we have metastasis or when the clinician says so. Okay? So to summarize, the outcomes evaluated with the standard approach, we can do every outcome, overall survival, progression, or clinical progression. I called it clinical progression-free survival, or whatever. The joint models can only do overall survival and clinical progression-free survival. We cannot do PFS because it's defined by SLD. Okay, a bit more information about the model of interest. For the longitudinal part, I just mentioned fixed and random effects. Let's dig a bit into it. I propose to have a second-order random walk for the average trajectory. So not the standard fixed effects. Random walk is basically an idea. The idea is to let the data speak. We have a smoothness constraint, but we will capture the average trajectory of tumor size conditional on treatment. So I really like it, but this is quite new. I developed it a month ago, so this is the first use, but it's very flexible, and it works quite well. For the random effects now, we kind of know what to expect in terms of trajectory. It's not so complex. Tumor size, usually it increases, it decreases, or it will decrease and then increase, U-shape or J-shape. So we don't need so much flexibility. We don't see some very strange curves in practice. So here I'm just including a random effect at baseline, which quantifies the variance in the tumor size at time 0, and then a linear and a quadratic term for the linear trend on curvature. This should be enough in terms of flexibility. So because the random walk is quite new and not many people know about it, even if it's quite an old thing, I propose also some sensitivity analysis with more familiar approach. Parametric functions, we just have fixed effects for everything. So fixed intercept, fixed linear slope, fixed quadratic slope, and then we have the same for the other treatment arm. That's more classic. And the splines, I'm using natural cubic splines with 2 bases. That's, again, enough to capture the trajectory we expect. And those are just to confirm that our random walk is doing well or not. Okay, let's discuss about the application. So metastatic colorectal cancer, a phase 3 clinical trial. The reason why I have access to this data is because it's made publicly available for research through the Project Datasphere platform. So the idea is they want to compare chemotherapy with chemotherapy plus panitumumab. Panitumumab is an anti-EGFR monoclonal antibody, and this is for second-line treatment. So we have 1,186 patients, but only 80% available, 854. The reason is for anonymization. They just share 80% of the sample, which is fine. Actually, it's good because we can compare our results to the full data results and see how well you are doing. So actually, the results are published. It's from 2010, so it's quite old, but they say panitumumab significantly improves PFS, progression-free survival, so it reduces the risk of progression, only among wild-type KRAS patients. So KRAS is a gene with two variants, wild-type or mutant. And this treatment only works among the wild-type. So we have this treatment effect modifier that we need to take into account. And they mention no significant difference for overall survival. So yeah, this gene, KRAS gene, many more have been found since then, but at the time, this was basically the main thing you want to take into account for those trials. So just a small description. First, we have the wild-type KRAS patients, and the mutant KRAS, approximately half of the population has one or the other variant. And for each one, we have the two treatment lines. And there are some covariates that we need to include because they were included in the original analysis. Those are prior treatment. It's second line, so they received the treatment already, and we want to adjust for that treatment. So those first two covariates are binary covariates for the previous treatment. ECOG is some performance score. It will basically say, is the patient stuck in his bed with symptoms, or is he feeling good enough to walk and everything. So it's also a binary, and it's an important predictor to include in the model. So we will include those three covariates, those three baseline covariates, in all the models, longitudinal, survival, just to match what is done in the original analysis. So the models are slightly more complex than I showed earlier because now we need to account for treatment and KRAS mutation. The interaction between those two will give us 4 trajectories. So my random walk for the average is actually a stratified random walk with 4 trajectories. For the quadratic and splines, I just need to include all the interaction terms between between treatment, KRAS mutation, and time. So that's it. We have also 4 average trajectories with the quadratic and splines. And of course, we can have baseline covariates. I didn't include them. It's already a mess, so whatever. So this is my evolution of tumor size over time for each patient in the trial, stratified by the KRAS gene. And the color is for treatment. What I see here is the so-called heteroscedasticity. What does it mean? Small tumors, you can see we have a lot of data here, very concentrated, small variance. Big tumors, it's more spread out, higher variance. Regression model, they want homoscedasticity. We need to have a constant variance. That's the assumption, usually. So what we can do here is a nonlinear transformation. And the most intuitive one here would be the log. And it has biological motivation. A tumor usually increases in size exponentially. So a log transformation here is a reasonable assumption. And I'm doing the log of tumor size plus 1 because there are a few zeros. It's advanced cancer, so only a few. But log of 0 is an issue. So I just shift by 1 unit, and then we are fine. So now you can see we don't have this issue with variance anymore. We sort of linearize trajectories. That's the idea. Let's look at the results. So first, I took 9 individuals from the data set, and I'm just looking at how my model fits their trajectories. So here you can see some trajectories decrease and then sort of linear, or some U-shape as we guessed. And the model is doing quite well. It do capture individual trajectories as we want, quite accurately. Now, on average, do we have a difference between tumor size over time, conditional, and treatment? Well, we do, as expected, because we know the results. So among the wild-type KRAS, we can see that the addition of panitumumab leads to a stronger decline of tumor size, and we have a significant difference between 6 and 15 months with the nadir at 1 year. So this significant difference is expected because we know this treatment should work for the wild type. And in the mutant, we can see no difference. As expected also, treatment has no effect for those guys. Just for the sensitivity, you can see with quadratic and splines, we get roughly similar trajectories. It's slightly more constrained. We need to have a very kind of smooth curve, while the random walk will be more like letting the data speak. So it's more flexible, but both are quite smooth. And we still see a significant difference here and here, especially in the parameters of the model. So we get similar results, very similar. Now, the association between tumor size and the risk of death or the risk of progression. This is quantified, but we don't really care. We care about treatment effect, right? So we need this parameter, but interpreting it is not a priority. And that's good news because on the log scale, then interpretation can be a bit tricky. We could interpret, but that's not a real interest. What we can see is those hazard ratios are always significantly higher than 1, meaning that a bigger tumor size is associated to a higher risk of progression or a higher risk of death, expected. The main interest is in those hazard ratios about the conclusion of the trial, right? So the standard approach, which is what they report in the journals, Cox model on PFS, 0.79, okay? We can compare it with the full trial because we have the results of the trial. So the full trial, we can see 0.73. We have a slightly stronger treatment effect with a bigger power because we have more individuals. Now, if we look at the standard Cox model on clinical progression-free survival, which we shouldn't look at because this is just overlooking tumor size. So this endpoint is for the joint model, but you can see that it shifts slightly towards the stronger effect, which is unexpected. I don't really know why. This is an artifact because we omit tumor size and we get a stronger effect. I would expect the opposite, but whatever. We don't really care. What we care about is the joint model. Here's the joint model. We increase the statistical power by adjusting on the heterogeneity, the individual heterogeneity. And we filter out the noise and mitigate attenuation bias, and we get what we expect, a stronger effect which aligns with the full trial. Of course, this is illustrative. You shouldn't be convinced by this. But we have the theory, and now we have an illustration exactly as we expect. It goes in the direction we want. So I will come back with a proof, but so far so good. It's looking good. We also have the joint model with current value, where we have a time-dependent hazard ratio now. We can plot it because it's not a single value anymore. And you can see we have evidence. The benefit seems not constant. It peaks at 1 year with a hazard ratio of 0.65, so stronger than the constant one, which average over the entire follow-up. And it fades after 18 months. So at the end of the follow-up, we don't have evidence for significant difference between the two treatment lines when it comes to progression. It could be because the treatment is not efficient anymore. It could be because we just don't have enough patients. Because over the follow-up, patients will die or be censored. So the sample size decreases. We have less and less statistical power, so we expect higher uncertainty here. And you can see the constant hazard ratios, they just average over the entire follow-up. So they mostly are influenced by the early data, which is associated to a stronger sample size. What about overall survival? As I said, the conclusion of the trial is no effect. So this is the original trial. They found 85.85 hazard ratio, but this is not significant. However, it still gives evidence toward a benefit. And when we compare the standard Cox model with the joint model, we can see that with a higher statistical power, we get towards the original trial, which has also a higher statistical power because of sample size. So that's also good news, but nothing much to say about it. Interestingly, the time-dependent hazard ratio here with the joint model, it shows some evidence toward the survival benefit. It even crossed the significant line at 0.05. I wouldn't consider this as a statistically significant difference. And you shouldn't, unless your name is Fisher, I guess, but otherwise, this is not good evidence, and we should take it as it is. We just have evidence towards the survival benefit around 1 year of follow-up, okay? And the constant hazard ratio, they average this out. Now, we had results in the application that aligns with the theory. Let's make a simulation study to verify robustness. So the idea of the simulation study is we have 3 models. Each model has a specific assumption about how the data is distributed. We can simulate data following those assumptions and verify how the other models behave when they are the wrong one. So for each simulation scenario, we have 3. I will generate 1,000 data sets, each including 1,000 individuals. The first one, the Cox, assuming it's true, the longitudinal marker does not add any information. No information about heterogeneity, no information about treatment effect. It's just noise, it's useless. What happens when we fit a joint model with a useless longitudinal marker? Second one, the joint model with shared random effect. The longitudinal marker informs about heterogeneity in the population, but it does not inform about treatment effect. Okay? And the last one, which is probably the more likely, we assume the longitudinal marker captures both heterogeneity and treatment effect. So let's look at the results. Out of transparency, I just show the how full results. It's a bit painful to read, but we don't need to spend much time on this. The idea is this is the first scenario, and we can see those are the parameters of the model. Those are the values I use to generate data. Those values, I choose them based on the application to replicate data similar to the original cancer clinical trial. I just increased a bit the sample size and the treatment effect, but otherwise it's very similar. So what we can see is we have bias in the first column, then standard deviation of the bias, and then coverage probability of the credible interval. How do we quantify uncertainty? Is it good or not? It should be 95% because it's a 95% interval. What we can see here is all the models are unbiased and give good properties. Good. We have those metrics, deviance information criterion. This is a goodness of fit metric. It allows to compare models. Does this model fit the data better than that one? Here you can see very residual difference. Given the standard deviation, one time is positive, one time negative. We have the same fit for the three models. They do the same. And if we compare the hazard ratio of interest, we get the same result. The constant hazard ratio, because the true model is the COX, so the true hazard ratio is constant. We also recover it with both joint models, especially the one that allows for time dependence. It still gives the same and with the same uncertainty here. Second scenario. Now, the longitudinal marker includes heterogeneity. So it informs about the noise in the population. This individual might be at higher risk, and we can capture it in the longitudinal marker. First, we observe what we expect, a bias towards the null for the treatment effect. This is treatment effect. It's a bit subtle because I have a lot of data in my simulations, but we still observe what we expect, a shift towards no effect, because we ignore heterogeneity. And the goodness of fit also, it shows this COX model is not doing well at fitting the data compared to the joint models. So if we compare the hazard ratio now, we can see the joint models are still doing fine, the hazard ratio is still constant, but the COX model is biased towards the null, as expected. So it shows what we discussed earlier. The last scenario, now the hazard ratio, the true one, is evolving over time because we share the tumor size over time, or the longitudinal marker. So not much to say on this slide, but I will directly go on the results of the hazard ratio. The true hazard ratio is evolving over time. And as you can see, it's beneficial in the early follow-up, but after 3 years, it crosses the line. It becomes a poison. So the treatment works well for a given amount of time, but after some time, you should stop it. It's not good. And the joint model with current value does capture this time-dependent effect of treatment, while the standard models, they average it out. And because we have more data in the early follow-up than in the late follow-up, here many people are dead or censored, the weight is higher here. So we will conclude the treatment is good, even if we have evidence for a deleterious effect in the late follow-up. So we miss some information. So to conclude on the simulations, the first scenario just showed that joint models introduce no bias. The second scenario shows that, as expected, the COX model is biased towards the null. And the last one shows that with the joint model with current value, we capture a possible evolution of treatment effect over time, and we can detect those late deleterious effects. Okay, so to discuss the joint model with shared random effects is heterogeneity adjusted, and it gives us an average treatment effect. The joint model with current value is also heterogeneity adjusted, but it gives a dynamic effect of treatment over time. One important point, we make proportional hazard assumption conditional on the longitudinal marker with a joint model. We compare big tumors with big tumors, small tumors with small tumors. So that's a very important point because proportional hazard sometimes is a strong assumption and it's very relaxed and more reasonable with a joint model. And as you can guess, beyond randomized clinical trials, this strategy to adjust for heterogeneity and imbalance is very appealing for observational studies where we don't have randomization. A significant barrier to the adoption of joint models has been the impractical runtimes of standard iterative or sampling algorithm. So that's why I spent the last 4 years during my postdoc to develop a new method based on the INLA algorithm, which is a very fast and efficient algorithm that was proposed in 2009. And I've been adapting a lot of biostatistical models to the use of INLA. So I developed this INLA joint R package, and you can see we have very reasonable computation times for the joint models, up to 1 minute 30, for the full application to my trial. So those 4 years of biostatistical development with INLA, they are summarized in a book that I just finished writing, and it will be published this year. And if you want to have an overview of the content of this book, I gave an invited talk at the University of Cambridge about a month ago, and they did record it and put it on YouTube. So if you want a 45-minute overview of the book, just check the video on YouTube. Okay, I think I'm done. Thank you.